I just want to confirm you guys all see the right part of my slides, not the part I don't want you to see. Okay, perfect. That's always a good start. And it's also nice that, Jeff was mentioning that how animal imaging has really exploded in the last 15 years. And I started as a postdoc with Jason Lurch in 2010. So it's about the right amount of time to see how all of this has kind of really evolved over the span of time. And it's nice to have you all here, and really looking forward to the feedback. I'm joined today by two senior PhD students in the lab. I'm going to give you a little teaser of their talks at the end of my talk. Stephanie Toulon and Gabriel de Jose Gregoire. And so I think definitely stick around for those talks, because they're the ones doing all the hard work, not me. And so I want to start a little bit with where kind of I started, which is really in human imaging. And one of the core emphases in human imaging, and one of the core goals has really been trying to understand brain architecture. And you can define that however you want, whether it be structural, functional, some integration of the two, how that changes over time and in terms of the lifespan. And more recently, trying to connect those changes to behaviors, genetics, or even cell types, or genomics in the form of transcriptomics. Of course, this is really hard to do. Most of this stuff is correlational. Obviously, you can't look at cell types in the human brain unless you have access to postmortem tissue, which is rare and hard to find. And that presents a huge opportunity in the context of translational imaging, in terms of animal imaging, where, of course, now it's no secret to many people in this call that you can get really nice assays of brain architecture, even though the mouse brain in our case, which is the species of choice, is very small, isn't folded, but it still has a lot of similarities, or you can develop similarities between the mouse brain and the human brain that you want to investigate. You can develop a lifespan context in a relatively short period of time. So from birth to adulthood takes about 90 days for a mouse. The gestational period is only 21 days. So there's a lot of opportunities there to look at drug development pipelines, environmental exposures, and so on, when you think about just the time span it takes for a mouse to become an adult or a mouse to get old. And of course, getting genomic or transcriptional information or cell type data, or developing novel genetic lines is ultimately quite doable in the mouse brain, in the mouse as well. And so a lot of the work that I'm going to be talking about today, almost all of it, has been occurring at the Cerebral Imaging Center, which is part of the Douglas Hospital and the Research Center, which is the main psychiatric teaching hospital at McGill, and on a fairly old now 17 Bruker biospec, but I think we do a lot with that technology still. And so, keeping up with the title of my talk, Improving Translational Neuropsychiatry, I think we do that in a lot of different ways. I'm going to spend a lot of time on lifespan context, what most people call sex differences, although it's arguably sex-specific research, and then a bit of a nod to what Stephanie will be talking about, which will be using MRI and small animal imaging in the context of treatment discovery, and a little of what Gabe will be talking about, and what forms a big chunk of his PhD, which is improved analyses pathways and standardization analyses pathways moving forward. So the lifespan context is where we're going to start. And so, like I said, a lot of brain imaging studies in the human are moving towards longitudinal assays. So if you think about the ABCD study, the idea is to have, translational assays, longitudinal assays, every 2-3 years in the same cohort of thousands of children over time. In the mouse, this is possible and tough to do. So here's some work that was been done by Lonnie Kupo, who's another senior PhD student in the lab. And this is work that we've done painstakingly over the last couple of years, trying to get dense sampling in a single mouse over the earliest phases of their lifespan. So, you can imagine that a mouse at postnatal day 3 is about the size of the top of your pinky. It's a little bit of a jelly bean. And if you just focus in on the cerebellum, you can see how the foliations of the cerebellum increase dramatically over the first 7 days of the mouse brain life. The increase in the frontal lobe, or the frontal part of the brain, increase dramatically over the first several days of life. And you're able to capture this, almost with the naked eye with MRI, if you're able to assay the same mouse over time. This has its caveats. But it is an opportunity to look at how, for example, in this case, brain development may be changing in these early phases. And of course, in our lab, we've come up with a number of pipelines, both longitudinal and cross-sectional, to model this type of change. These aren't, they're definitely optimized for the type of scans that we get, not necessarily novel in the context of longitudinal brain imaging of what's been happening in Canada, but certainly something that we've been working on very hard. We all know, and this is something I'm going to allude to later on in my talk, when you start doing a new project in animal imaging and small animal imaging, whether it be on the acquisition side or the post-processing side, everything's new. And there's a lot of work that goes into just optimizing the acquisition and then the subsequent data analysis pathways post-hoc. And that's something that we've been working on to make more robust in our lab. But in our case, in these dense longitudinal samples, or any longitudinal sample, really, we can model growth or change in a signal brain over time. And this is something I'll be coming back to over and over again in the talk, where we use nonlinear registration to create subject-specific averages that are sensitive to within subject change over time, which are then warped to a consensus average where we do our statistics. So in this particular example, one thing or in general, what we want to do, we've been applying these types of methodologies to a lot of different problems in the lab. And this is something we've been doing kind of from a technology development standpoint and from a scientific standpoint from across the lifespan. So one thing that Lonnie's been working on very hard, and she's at the end stages of her PhD, is this, these open problems in almost population level science, where we've been asking the question, what does cannabis exposure look like in the first few days of gestation, and what are the long-term outcomes on offspring? And this is really important because, as we all know, cannabis has been made legal in Canada for recreational use, and this is just the adjusted prevalence of pregnant people using cannabis in the first trimester of pregnancy as a means of coping with issues related to anxiety, depression, and nausea, and so on, right? And you can see that prevalence of cannabis only going up across the U.S. And I don't think there's any reason to think that Canada would be any different. And so when you expose animals or moms to cannabis, to THC in this case, which we inject in the earliest phases of life, what you see are these subtle changes in brain development that if taken cross-sectionally, if you looked at any of these one time points, would almost be imperceptible. But it's this change in growth rate where you see this decelerated or growth in the pups that is really telling. And it's something that we've been investigating more and more. So I'm just using this as an example of the type of things we can do with the strategies that we've created. But what we've really been trying to do kind of on top of that is try to create these lifespan trajectories in different contexts. So this is, and one of the things we started working on in the context of brain development, but also in terms of long-term outcomes, is look at the impact of the inflammatory state of the mother during pregnancy. And this is important because now it's important because of COVID, and I wish I could say we started this right on the heels of COVID starting, but we actually started this a few years before the pandemic. And what we do know is that the immunological milieu of the mother during pregnancy is a really, really important indicator of outcomes in the offspring. And increased levels of inflammation, whether it be due to illness, bacterial infection, obesity, or even underweight mothers undergoing famine, really act as a risk factor for a whole array of disorders. And these include disorders that occur during the gestational period, and the genetic vulnerability of the parents. And what you often see in individuals exposed to inflammation during pregnancy is a heightened risk for neurodevelopmental disorders that include psychiatric comorbidities like autism, schizophrenia, and higher susceptibility to second hits induced by drugs or stress later in life in vulnerable periods like adolescence. And finally, in adulthood, you see a heightened risk again of psychiatric and neurologic disorders, including things like schizophrenia and depression. And so we wanted to ask the question, okay, given that we know that the lifespan of the mouse is very short relative to ours, and doing investigations that involve brain imaging like this in humans is really challenging, can we now model this in a mouse model? and try to stitch together a lifespan of what inflammation might look like, or the response to inflammation might be. So what we've been doing is modeling prenatal maternal immune activation in mice. And we do this using poly-IC, which is a double-stranded analog of RNA. And when injected in the mouse, it creates this pro-inflammatory response. So there's a elevation in pro-inflammatory cytokines. So it basically fools the immune system to think there's an attack on the immune system, which crosses the compartment from the paternal compartment. When injected in the mouse, crosses over from the maternal compartment through the placental compartment and into the fetal compartment, and may also have effects on the ordering and the organization of the placenta itself. And so this was work that was carried out by Elisa Guma, who's about to start a position at Harvard, actually, in the next little while, related to the immune state and the neuropsychiatric disorders. So very much in keeping with the work that she did in her PhD. And I've highlighted a couple of the papers here that she published, but there's a series of 4 of these from her PhD that are really worth looking at if this is something that you're interested in. And so in this case, what we could do is we could start asking the question, can we systematically tease out what the impact of tampering with the immune state of the mother might be? And so I've made this is the timeline of brain development from conception to adulthood in what would be a human. And what we wanted to do was mimic 2 specific gestational epochs. One, which would be more or less late first trimester, early second trimester in the human, which translates to gestational day 9 in the mouse, and gestational day 17, which is but late second trimester, early third trimester in the human. And you can imagine that there's a lot of literature on the timing of the inflammatory insult and what that might do to the offspring down the line. But you can imagine, if you just look at this kind of very crudely, that something that happens earlier on the gestational period, such as gestational 9, which is what we're going to call early life inflammation or early life exposure for the rest of this talk, may tamper with neurogenesis or gliogenesis, whereas some of that happens a bit later on, such as in GD17, may tamper with neural migration. And so we did this this is one example of an experimental timeline where we did this in dams using the 2 different timings. And in this case, just to look at what's happening during the fetal development process, we extracted embryos at GD18 and scanned them ex vivo using MRI. And we did this in collaboration, in this particular case, with Brian Nyman and Jason Lurch and Shoshana Spring at the Mouse Imaging Center, using, I guess, almost now what is out-of-date technology for them. And they've moved on to other better technologies at Higher Field for doing simultaneous postmortem imaging. But we got images at 40 microns, and using something similar to the pipeline that we mentioned before, you can create kind of an average representation of the anatomy that looks something like this, which is a beautiful representation of not just brain, but also body of the brain using pipelines that we adapted. And when we do that, you can model the changes in the differences in volume, in the GD9 mouse. And what you see is that you have altered brain volume when exposed at GD9, when the inflammation is introduced at gestational day 9, and you harvest the embryos at gestational day 8, compared to a saline injected control. And these changes in volume occur in interesting places like the prelimbic area, the septal nucleus, subventricular zone, the caudate and the putamen, hippocampus, dentate, gyrus, and the cerebellum. And so, by and large, you see some increases in volume, some decreases in volume. But what's interesting is that now, because you have the embryos and you have the tissue, you can now harvest, or you can look at cell types, for example, like I was mentioning before. And we did this in collaboration with Maud Bordelot and Marie-Ève Tremblay, who's at University of Victoria. And we looked at a number of different markers of kind of cellular stress. And so here we used electron microscopy and achieved, nanometer scale resolution in the postmortem tissue. And we did these experiments in dorsal hippocampus. This is where we saw differential effects of MIA almost throughout all our experiments, which I'll show you in a little while. And we looked at the density of dark neurons and glial and apoptotic cells as putative markers for neuroinflammation and oxidative stress. And we saw that mostly in the mice that were exposed early, you see higher instances of these apoptotic cell densities. While the late group seemed to have, to a certain point, a greater density of dark neurons and dark microglia, although these weren't statistically significant. And also the samples are quite small here, given the labor involved in scanning these. But this gives you some indication of what might be happening under the hood, which confers risk for later in life. And so if we go back to this idea of being able to stitch together a timeline and of the whole lifespan, you can now start thinking about what happens at GD18 in the embryonic stage and how early and late exposure may differentially impact the anatomy of the brain. Which leads us to then ask, okay, what's happening later in life? So early on, neonatally, and then stitching together this timeline from childhood to adulthood. And so using a very similar experimental design, but letting the mice get older, we now assayed brain anatomy from PNG21, so roughly when the mice are weaned, over 4 time points all the way to adulthood. And now you can stitch together these timelines of change that are happening anatomically at the single mouse level, and then ask the question, how are the rates of change differing over time? And so here are just some canonical curves that we saw. What we generally saw is that those offspring that are exposed at GD9, so with poly-IC early, have this little undershoot here and have a bit of an overshoot on volume, and then have this normalization in volume that looks a lot like the saline controls and the late exposed animals. Overall, we see a lot more kind of severe changes in these early exposed offspring. So that's something else that's quite interesting, and maybe indicative of vulnerability windows for this type of environmental exposure. We see the same thing in the cingulate cortex, the striatum, as well as the hippocampus and the dentate gyrus. So now, using this and some other multivariate methods where we connected some of these phenotypes, which were quite similar to patterns of brain and behavior, which I'm not going to show, what we were able to do was then examine some of these regions as they relate to the transcriptomal profile, of some of these regions, sorry. And we did this in collaboration with Rose Baggett, her postdoc Vedrana, and Pedro Bourguignon, who used to be a research assistant in that lab. And she helped us run a number of RNA sequencing experiments where we harvested tissue from fresh frozen tissue from animals after GD9 exposure in a number of areas that we saw to be affected. So like I said, the prefrontal area, the hippocampus, as well as the dorsal and the ventral hippocampus. And what you can find here is on these volcano plots are upregulated and downregulated genes that are upregulated and downregulated in these areas after GD9 exposure. And by far, you can see very clearly that the dorsal hippocampus had the largest preponderance of differential gene expression. And so if we kind of look at the neuroinformatics analysis of that gene expression after the fact, just in the dorsal hippocampus alone, I'm going to show you kind of, sort of showing the genes one by one, I'm going to show you the results of the enrichment analysis. So here again, upregulated kind of pathways in red, downregulated pathways in blue. And first of all, the dorsal hippocampus, you see significant enrichment for FGF signal, which is classically involved in tissue repair, but is a critical signaling pathway in embryonic brain development. And signals for the patterning of the spinal cord, supports normal gyrification, synaptogenesis in the hippocampus, amongst other really important functions in brain development. We also see an enrichment for autistic behavior, very much in keeping with our initial hypotheses, and microRNA regulation, which are small non-coding pieces of RNA whose dysregulation has been associated with a number of brain disorders, including psychosis and depression. So all in all, this paints a picture of how we can now use these type of assays to kind of get some insight into what might be happening in the human condition. And then we can go back to our timeline and start stitching together the different pieces of what might be happening. So, for example, the overshoot and the normalization in the early exposed mice, followed by the transcriptomal profile. There's a homologous story in the late exposed mice that I'm not providing here. And there's some experiments that we did in the early lifespan, which showed very subtle differences in anatomy and some disordered behavior that I'm not showing here today. But this gives us a sense of how where the vulnerability in windows might be. and how they may be impacted by the different types of exposures that are seen prenatally. I'm next going to talk about sex differences. And this is something that's kind of a keen interest of mine and a keen interest of our lab. I think a clear need for, and something Stephanie will talk a little bit about in the context of her project later on. In this particular case, we've been looking at sex differences probably in a disorder that really needs sex-specific investigation, and that's depressive disorders. It's no surprise that it's one of the top causes of disability worldwide. It impacts at one time or another 4% of the world population, but it's almost 1.7, if not more, fold greater incidence in females compared to males. And what's also interesting is that the symptom profile in females, in severely affected females, is also different than the symptom profile in males. And that, I think, is something that really needs to be investigated at the level of animal models. And so if you look at models of stress, one very common one is this model called chronic variable stress. And it's basically exposing the mice to a different environmental exposure, stressful exposure for an hour a day. This includes foot shocks, tail suspension, and restraint, and you cycle through continuously daily until you get, until a stressful response is encoded, and then you get a depressive-like phenotype. What's really interesting, despite the differences in stress response in males and females, and despite the differences in the symptomatology, prevalence, what you see over and over again is most labs use male mice for this specific task, for this specific exposure, and there are very few labs that have investigated females or males and females together. Now, there's a number of reasons for this, not only the well-known bias towards using males in rodent research, but also different exposures, different levels of exposure to the stressful to these stressors give different outcomes in the animals. So in females, just 6 days is enough to create a depressive-like phenotype in these animal models. But to get a similar level of a depressive phenotype, people have investigated 6 days, 15 days in males, and you don't actually get much of a depressive phenotype as indexed by sex-specific translational profiles, hormone-related mechanisms, and synaptic mechanisms in the limbic circuits, for example, which are well-known kind of phenotypes related to a depressive signal. And you need up to 28 days of exposure, daily exposure in males to get something that looks like a depressive-like phenotype compared to the females. So this is work that was done, that's ongoing by Liz Herrera-Portillo in my lab, who's also a senior PhD student, and in collaboration with one of my strongest collaborators, Rose Baggett, who's at the Department of Psychology here at McGill. And I'm going to walk you through a work example of what we did in the females, then I'll circle back into the males and show how we can get sex differences type results in this type of investigation. And so when we expose animals, females, to 6 days of chronic variable stress, you start seeing that they start showing an anxiety-like phenotype as indexed by the amount of time spent in the center on an open field test. And they also show differences in social preference, which is another anxiety and depression-like phenotype compared to the non-stressed counterparts. But of course, there's a huge spread in this data. It's not always conclusive. There are mice that more heavily respond to the stressor than others. And so we need ways in which to look at this. And so, of course, MRI, like we're all here for, is one example. And if you do just a univariate analysis on this, and this isn't related to the behavior at all, but 6 days of stress in the female mice is enough to create really significant neuroanatomical remodeling in female mice. And this includes regions that are very much part of stress circuits like the hippocampus, the hypothalamus, the amygdala, the accumbens, for example. And so what we wanted to do is we wanted to create kind of a spectrum of disorder neuroanatomically and behaviorally based on what we found to see, are there ways in which we can model the data that is a bit more akin to what is reality. Instead of cherry picking one or two behavioral tasks, Can we use the whole array of behavioral data that we have? Can we use all of the neuroanatomical data that we have? So what we did is we scanned the mice pre and post-stress, kind of in early adulthood. And we used a technique called partial least squares, which I'm not going to go into the details of. I imagine many of you are quite familiar with it. But what it looks for is patterns of covariation that are orthogonal to one another, that are encoded as latent variables. So in a single latent variable, you get a pattern of covariation of positively correlated and negatively correlated voxels, which is what you see here, that are bound to a behavioral pattern, in this case, using the measures that we have from the open field test and the social preference test, which indicate both the anxiety-like and social dysregulation that these mice undergo. And for each of these mice, you get a score of how much they load onto the behavioral pattern and how much they load onto the brain pattern. You can plot that into dimensions now and summarize the data that you have. And what you see is that there's a clear separation between the stressed mice, and the control mice, the unstressed mice, in this kind of new latent variable space. And so you can use this kind of multivariate technique that's well used in the human literature to kind of find what are the dimensions of disorder and dysregulation in our animal models. So Liz was curious about this, and she wanted to look at how these positive volume changes that encompass a really interesting set of areas, the lateral septum, the nucleus accumbens, hypothalamus, and amygdala, a negative volume change in the insula anterior cingulate that are now correlated to increased anxiety-like behavior and decreased social preference. How are they ordered? How are they connected? And how can we use a connectivity profile between these areas to understand more about the organization of these areas and why these might be preferentially changing both in the positive volume sense and the negative volume sense? And for that, what she did is she took the brain pattern from the partial least squares analysis and chunked it up into the positively correlated volumes, voxels, and the negatively correlated volumes, assuming that they, under the assumption that they represent two different processes that are going on simultaneously. And what we did is we used the data from the Allen Brain Connectivity Atlas, which is a series of injection experiments using 491 antegrade spiral tracings in the C57 mice, which is what we use to trace out monosynaptic connections across the whole brain. And what we did was use a high-resolution data-driven model of the mouse connectome to better to use as kind of a backbone, a scaffold of the connectivity anatomically of what might be going on between these regions of between these regions that have changed together. And so what we can get is that we can use any one of these blobs, as we'll call them, that are connected voxels that are separated from the other blobs as an ROI and look at the connectivity from that ROI using the Allen Connectivity Atlas to the rest of the brain, and take basically this, like you would with DWI, but in this case the tracer experiments, as regions that are connected to one another. And you can create this adjacency matrix or this directed structural connectivity matrix and threshold it for significant connections. And that's something she did using a laborious permutation test. And so when you do that, you can then create a graph-like structure where the nodes now become any of these connected blobs or connected regions from the positive or negatively correlated regions, and the edge is a monosynaptic connection. You can then look at the community partitions within these regions, so the number of connections within a community. A number of connections within a community is usually greater than a number of connections between communities, so you kind of create segregation of blobs or of these regions. And we look for rich colors, for the tendency of highly connected nodes from these interconnected regions, which are thought to promote efficient communication and functional integration across brain regions. And so if we do this, so this gets pretty hairy in the next couple of slides. There's a lot of data, but I'm going to try to summarize it in the end in a more digestible format. So what we have here now are the communities that we detected using our graph theory method, and the nodes are representing kind of the different parts of the anatomy that are in the cerebellum, the hypothalamus, the thalamus, cortex, striatum, pallidum, etc. We have this kind of in the negative correlation network and the positive correlation network. And from this, we can now, again, from the community information, find parts of nodes that have a high participation coefficient, which are thought to really represent regions that are involved in functional specialization or maybe vulnerability towards the depressive exposure, the stressful exposures that we showed before. The other thing we found were a bunch of nodes that form a rich club core. So it's a core of nodes with a high number of connections that are more connected to one another and more connected to their overall communities. So basically, they're thought to pass information between different communities within the brain and promote efficient communication and functional integration across brain regions. And we have one of these for both the positive network and the negative network. And what I'm going to do in the next slide is kind of show you kind of a network structure, kind of a model of communication that we think is happening in the females after just 6 days of exposure to this stressful phenotype. And that includes the thalamus, the amygdala, the lateral septum, the hypothalamus, the ventral tegmental area, and the ventral striatum for these negative correlations, and different regions, visual cortex, anterior cingulate. The thalamus participates in both, and the dorsal striatum, so a different part of the striatum, participates in this kind of positive correlation. So what we found in the females is a high segregation of communities, a network of highly connected regions, negative and positive volume change, which are associated with depressive-like behaviors, and a greater number of vulnerable blobs to insults. What we can also do is qualitatively at least assess how different these are from the male condition. So on the negative correlation, we see generally some regions that are similarly represented in this rich club connectivity. However, for example, there are key differences, such as the hypothalamus being implicated in females and the hippocampus being implicated in males, both of which are regions that we know that have been reported to be heavily connected with depressive-like phenotypes. If you look at the positive correlations, the structure is very different. There are fewer regions involved. And for example, the insula is highly involved in the males and not in the females. So this further motivates kind of better and more sex-specific investigation so we can get a better handle on sex differences and disorders that need them. So finally, I'm gonna talk about two more things, both that are involved with translation in the 5-6 minutes that I have left. And one that, I mean, Ravi's very heavily involved in, which is the use of this type of machinery in the context of treatment discovery. And one thing that we've been really fortunate to be part of is this broader network, which I'll talk about in a moment. But our goal in this network is to really use the machinery that I talked here about being able to correlate brain and behavioral phenotypes in a very critical phase of the drug discovery pipeline. And this is this preclinical phase where many compounds are produced, but this is kind of like the final screening phase before you get to this 10-year-long cycle of clinical trials. And the premise of what we're doing is we think that, one, it'd be great if we could screen failures very early on, so you don't waste years of time, money, and human capital in these clinical trials if a drug is likely to fail. The second thing we want to do is be able to be more cost effective when you get to this phase, so you have an idea of what may work and what might not work. And so right now, what we see in kind of the preclinical space is that there's behavioral tests that are relevant basically for the animals, and you know, to be honest, a lot of the behaviors that I showed earlier on. So for example, you think of the open field test, right? We never ever put a human in an open field and ask them and see how they behave. And so how that really translates to human is something that we're all comfortable with, or many people are comfortable with in the animal community, but many are not. You know, there's no real easy translational kind of pathway to humans. There's no widespread use of mesoscopic imaging, and we often rely on postmortem pathology as the readout for neurodegeneration. And the solution that I'll tell you who we is in a moment, have been thinking of, are using reproducible methodologies, automated, high throughput and standardized, using imaging that is relevant to humans, so MRI, and may serve as a biomarker of toxicity, using cognition that is relevant to humans and may serve also as a biomarker of toxicity, and do this in a way that we can champion reproducibility by making this multi-center from the beginning. So instead of me doing the last two experiments like I showed you just in my lab, when you think about drug discovery, really being able to champion this as something that should be happening across sites. And so this opened the door to a large-scale initiative that is funded by two CIFREFs, one at Western and one in Montreal, one at McGill, Brain Scan and the Healthy Brains for Healthy Lives initiative, where together with Marco Prado, we've put together a really impressive team of investigators, students, and trainees who are seeking to work on this as a translational pathway. And this forms one of the pillars also in an initiative that Ravi's leading from the NFRF, known as Trident, which I suspect he'll be talking about at some point in this meeting. But Stephanie, who's going to be speaking next, is a big major player in this initiative, and a lot of the work that she did early on paved the way for what we're doing. So what this initiative focuses on is trying to find drugs that will treat neurodegenerative disorders. In this case, the focus for Marco and I is on Parkinson's disease, PD. And we know that the symptom profile that is often seen in PD, so stoop posture, the tremor, the rigidity, is because of neurodegeneration, often in the substantia nigra, but elsewhere in the brain, that results from the traveling of alpha-synuclein up from the brainstem into the brain and through the brain, causing toxicity and ultimately neuron and cell loss. And this was first indicated by the Bracken-Brack staging that is very popular in the field and the human literature. And so what happens with alpha-synuclein is that it may accumulate within neurons, cause Lewy bodies, and there's synaptic transition from cell to cell in a prion-like manner, which has been shown both in cell culture, in animal models, and in actually human experiments where there have been grafts of stem cells. And so what we've been doing is we've been looking at the M83 transgenic mouse, which is expressing the human form of alpha-synuclein under the control of a mouse prion promoter. And this is work that was pioneered, like I said, by Stephanie Tillo, and she's been doing this in wild-type mice and M83 hemizygous mice in our lab, where she injects preformed fibrils of human and sometimes mouse alpha-synuclein into the brain and looks across time at how pathology may be present at 30 days after injection and 90 days after injection. And typically you see symptom onset around here, around 90 at 120 days. And what that looks like is, this is Steph's video, is you have, oh, Steph, I'm not stealing your thunder, but these are two mice. One that has received the injection. Oh, this is maybe slowing down, but what you're supposed to see is a reduced mobility in this mouse, reduced rearing and grooming behavior in this mouse, compared to the mouse that has been uninjected. I don't know why this is not showing. Who will be able to rear on its hind legs and be able to look for, be able to rear on its hind legs. I don't know why this isn't playing. I apologize. Ah, here we go. I'll show you, maybe fast forward here. So for example, when we take the bedding out of the cage, sorry, I don't know why this is slowing down, but it is. I'm going to move forward just in terms of time. And so what Stephanie did in a cross-sectional experiment, which is now published in the Journal of Neurochemistry, that's 2023, she used a multivariate technique similar to a PCA analysis to look at network-like spreading in the mouse and finds this canonical signature, a biomarker, if you will, of neurodegeneration in the mouse brain after you inject with these preformed fibrils, suggesting that even if you inject in the dorsal hippocampus, you get a spreading across the brain that gives you a neurodegenerative pattern. And the neurodegenerative pattern we see is elevated in the mice that received the preformed fibril injection and is akin, at least qualitatively, to what we see in other human experiments using similar techniques and that show different regions. And so we've also using touchscreen tasks, using the Bussy-Succeeda touchscreen methodology, where a mouse is exposed to two different stimuli and goes and does a nose poke and learns to associate the correct shape here that it pokes on with its nose with a milkshake reward back here. And then that task becomes harder over time because the association with the reward is reversed. And what we find is very early on, before we even see motor deficits, you see a cognitive deficit and their ability to reach accuracy at an early time point in this touchscreen, after receiving the preformed fibrils. And by the time it's quite late, 85 to 95 days post-injection, you see a very big cognitive deficit. So this suggests that these two things can be looked at as biomarkers in this context. And one thing we've been doing is looking at all sorts of potential therapeutic interventions that might, where we can use these biomarkers and use them as things that can, and observe their reversal in the context of drug discovery. And we've been focusing very much on the chaperone and co-chaperone machinery, which is expressed in normal cells and cells undergoing pathological processes related to aging, genetics, and protein misfolding. But they create an epichaperone when they dysregulate, and then they sequester proteins and are thought to be involved in promoting the misfolding of alpha-synuclein that occurs in these animal models, and in the human condition as well. And so recently we worked with Marco on a paper where we use these biomarkers to look at a potential genetic target, which is STI1, which overexpresses some of the heat shock proteins, which are known to be involved in the proper functioning of the chaperone. And what you can see is early on, mice that overexpress STI1, that are heterozygous for the genotype, they don't have the behavioral deficit that I showed you before. And overall, if you just kind of focus here, their neurodegenerative signatures are basically gone, absent, compared to wild type animals and M83 heterozygous mice. So suggesting that these signatures that we have may act as biomarkers for possible therapeutic, for discovery of novel therapeutics in the context of this, but also down the line, we're looking at other models related to Alzheimer's disease as well. So finally, I think this really sets the stage. I only have 3 more slides left for really improving analysis pathways, like I mentioned earlier on in my talk. So, and recently, in work that we did with Joe Grandjean, and this is a really great resource paper that was published in Nature Neuroscience this year, and had a really, really strong involvement from Gabe Desrosiers-Grégoire, who's going to be talking to you later on. This represents our kind of first foray into the functional imaging space, and this is something that we've been really actively pursuing, and Gabe's been working really hard on, and you'll be hearing a lot about that later on. But if you just look at what people do in rats, for example, just in terms of what they do in terms of anesthesia, you see so many different cocktails that are used in just sedating the mice, so many different field strengths and so on. And so Gabe was tasked at the beginning of his PhD to look at fMRI in mice and how to use it as a tool for scientific investigation. And as he went down that path, what he found was over and over again that there was no standardization in the space. It wasn't clear what data quality looked like. And I urge you all, if you're interested in this space, to have a look at Gabe's pipeline and Gabe's preprint, which we're about to submit for publication. And he, through, I think a labor of love over the course of many years, has created the software pipeline that's used by many, many people around the world, called Rabies, Rodent Automated Bold Improvement of EPI Sequences. It's had over 2,400 downloads. Its robustness has been tested over data from across 60 different sites from around the world. So it's a really, really important, I think, contribution to the scientific space in this milieu. And the goal is to provide several reports that can add as scan level quality markers, provide scan and data level thresholds for quality, provide group level statistical reports on the quality of your data and what motion and other physiological measures may be doing to your data, characterize these issues and standardize reports that can be used as outputs that can be included in publications, and then to help people improve quality and optimize confound correction and adapt analysis and improve their acquisition protocols. So I urge you to reach out if this is something that you're interested in. And Gabe won't be talking about this today, but he will be talking about his ongoing quest to improve analysis in fMRI and in rodents. So with this, I'm going to conclude, and I hope I've conveyed that we think there's increased benefits of using translational assays like neuroimaging and of course cognitive tasks to gain insight into psychopathology and pathophysiology in rodent models. We need better links to their genetics, environments, phenotypic expression at the individual level to really understand their ultimate impact on psychopathology. And we think that these are key ways forward to using basic and clinical neuroscience to parse heterogeneity and ultimately promoting precision medicine. So I'd like to thank all my collaborators, and of course my son, Pinaki, who puts up with weird hours and me going to weird places to do things. Sometimes he gets to come along to Paris and sit on the Grand Roue and then I make him sit 2 days in a conference, in the Sorbonne or something like that. So, give and take. And he's a really good sport with all this stuff. And I really appreciate that. And of course, all my collaborators, both locally at Western, at UVic, Oxford, and SickKids. My funding, obviously, and of course my lab, without whom none of this would be possible. So I appreciate your attention and many thanks. I'm really excited to hear your questions.